You'd think by now we have a pretty complete map of the human body, right? I mean, we've been studying it for centuries. Well, today we're diving into a story that proves our own cells are still hiding some pretty big secrets, including brand new structures that have been there the whole time. So let me ask you this. Think back to high school biology. You learned all about the cell, right? The nucleus, the mitochondria, all the usual suspects. It really seems like we've got it all figured out. But what if that map was incomplete? What if a major piece, a whole new organelle, has been hiding in plain sight this entire time? All right, so our story kicks off with this huge announcement, this incredible discovery. But right out of the gate, there was a little bit of a problem. A puzzle that was all about its name. So, in 2025, a team of researchers announced something that really shook the world of cell biology. They had found a totally unknown organelle inside our cells, and they gave it a name, the hemifusome. This was a massive eureka moment. But almost right away, anyone familiar with fruit fly genetics probably did a double take. Because, you see, the name hemifusome, well, it wasn't exactly new. It had been used for decades to talk about something completely different. So what was going on here? Okay, so let's clear this up. Turns out we're talking about two totally separate things that just happen to share a name. On one hand, you've got the original fruit fly hemifusome. It's a temporary little bridge between egg cells. But on the other hand, you have this bombshell discovery in mammals, a brand new, permanent organelle that does something completely different. And that new hemifusome is what this is all about. So the big question is, how was this even possible? I mean, how did a structure this fundamental stay hidden for so long? Well, the answer isn't in what we were looking at, but how we were looking. It all comes down to the technology. You see, for more than 100 years, the tools we used to peek inside cells had this one major flaw. The preparation process was harsh. It involved all these chemicals that would basically destroy the most delicate structures before we could even get a picture. The poor hemifusome was just too fragile to survive. And this is where the breakthrough happened. The old way of doing things would just cause these delicate things to fall apart. But cryo-electron tomography, or cryo-ET, is a complete game changer. It flash freezes cells in a fraction of a second, locking everything in place, perfectly preserved in its natural state. It's literally like freezing a moment in time, letting scientists finally see these things as they actually are. Okay, so now that we can actually see it, what in the world is a hemifusome? What does this brand new piece of our cellular machinery really look like up close? Okay, so at its heart, a hemifusome is made of two different vesicles. You can think of them as tiny little bubbles inside the cell. But here's the wild part. Instead of fully merging together, they're kind of stuck, halfway fused, sharing a stable wall between them. This state, which is called hemifusion, was always thought to be this super fleeting, millisecond-long event, not a stable, permanent organelle. Let's break it down. You've got a smaller, clearer vesicle, and a bigger one that looks more grainy inside. That wall between them? That's the hemifusion diaphragm. But honestly, the most fascinating piece of this puzzle is this little particle sitting right where they meet, something called the proteolipid nanodroplet, or PND for short. And get this, that little PND isn't just some random speck. It is incredibly consistent, always measuring almost exactly 42 nanometers across. Now, to give you some scale, that is about 2,000 times smaller than the width of a human hair. That kind of precision in biology is almost never an accident. It's a huge clue that this particle has a very specific and very important job to do. So, you're probably thinking, a newly discovered organelle must be incredibly rare, right? Well, that's what makes this whole thing so mind-blowing. The researchers found that these things make up nearly 10% of all the little vesicles at the edge of the cell. They weren't just hidden, they were hiding in massive numbers right under our noses the whole time. And if you're trying to picture all this, the researchers came up with a really perfect analogy. They said it looks kind of like a snowman wearing a scarf. The two vesicles are the snowman's body and head, and that mysterious little PND, that's the knot in the scarf, holding the whole thing together. So we've seen what it is, but now we get to the most important question of all. What does it do? Because this is where the discovery goes from being just interesting to being truly revolutionary. For decades, we've had this textbook model for how our cells build their internal recycling centers. They're called multivesicular bodies, or MVBs, and they're absolutely critical for cleaning up waste. Well, the discovery of the hemifusome basically throws a giant wrench into that established theory. So here's the main difference. The old model, the one in all the textbooks, is called the ESCRT pathway. It's all about proteins. It uses this complex crew of proteins to do the construction work. But the hemifusome points to a totally different way of doing things. It suggests there's a second, ESCRT-independent pathway, one that's driven more by lipids and physics, using that mysterious P&D to reshape things instead of a big protein construction crew. So what's the hemifusome's job in all of this? The best way to think about it, as the researchers suggest, is like a cellular loading dock. It's a platform where cargo can be sorted and packaged up, creating an entirely new system for managing all the traffic inside our cells. Now, a discovery this fundamental doesn't just stay in the lab. It immediately blows open entirely new ways of thinking about our health and about disease. And this, right here, is why it matters so much. When this cellular recycling system breaks down, the results can be just devastating. This new pathway could be the missing piece of the puzzle for understanding diseases like Hermansky-Pudlak syndrome, which is directly tied to these recycling centers. And it could even give us new insights into neurodegenerative diseases like Alzheimer's, where clearing out toxic junk from the cell is a huge problem. Ultimately, discovering a whole new cellular pathway is like finding a new door into a room we thought was sealed shut. It gives scientists a completely fresh perspective to figure out what's going wrong in these incredibly complex diseases. And who knows, one day it could lead to entirely new ways to treat them. And that just leaves us with this final thought. The hemifusome was there the entire time, a basic part of our cellular machinery, just waiting for us to invent a tool that could finally see it. It really makes you wonder, what other secrets are our cells still keeping? What else is hiding in plain sight?